The overall goal of the following experiment is to functionally characterize extrinsic afferent nerve endings in the mouse colorectum in vitro. This is achieved by harvesting the colorectum and the attached pelvic or lumbar splanchnic nerve in continuum and placing the tissue in an in vitro recording chamber. As a second step, the pelvic or lumbar splanchnic nerve trunk is split into fine filaments so that single fiber electrophysiological recordings can be made of individual nerve axons. Next, individual colorectal nerve endings are identified by unbiased electrical stimulation of the receptive fields and functionally classified based on their response profiles to 3 distinct mechanical stimuli. In addition, responses to chemicals applied to the nerve endings can be tested by isolating the receptive field with a piece of brass tubing and replacing the solution with the solution containing the chemical or mixture of chemicals. Results show that mouse colorectum is innervated by 5 different classes of mechanosensitive afferents. In addition, about 25% of colorectal afferents are mechanically insensitive or silent nociceptors.
This method is used by us and others to study the sensory innervation of the colorectum. We're interested in using this research approach because many gastrointestinal disorders are associated with sensitization or a change in the excitability of the sensory innervation. Our interest in understanding the mechanisms of sensitization relates to our desire to help develop therapeutic strategies that then might be useful.